So our final talk in the climate change session this morning is going to be a little bit different. We're going to shift over to talking a little bit about plant and pollinators. So Laura Lampe completed her master's degree in biology from Chico State in the fall of 2020. She has worked as a field botanist and wildlife biologist in consulting and managed the gardens of Chico State's Gateway Science Museum, home to nearly 200 native plants for over 4 years. As a museum creator, she also recently participated in the production of the museum's current exhibit, Bombus, A Natural History of Bumblebees, which she co-curated with Jennifer Jewell and Jason Whittlesley. Laura is here to share the results of her master's research, which explored the long-term collection records, what long-term collection records reveal about phenological misalignment between Northern California plants and their pollinators. So welcome, Laura.
Hello. Thank you all so much for having me here. What a dream. It's so nice to see everyone and see some people that I haven't seen in a while. I'm really stoked to talk to you all today about my master's research, which I wrapped up a couple of years ago, or a year and a half ago. Time is wild. But I'm going to be talking a little bit about how long-term collection records reveal phenological misalignment between Northern California plants and their pollinators. And oh no, nobody told me how to change the slide. Do I just press the, oh no. Oh, there we go, okay, maybe? Okay, awesome, we're there. I'm going to start out with a little bit of background, which I'm sure many of you are familiar with, just about some of the main concepts that we looked at in this study. So we know, of course, that climatic cues drive phenology, which is the timing of life history events, such as blooming and germination in plants and emergence and activity periods for pollinators. We know, for example, that factors like the timing of snowmelt, soil moisture, and temperatures can prompt plants to germinate and prompt insects to come out and emerge and be out and about and able to pollinate. We also know, of course, that when plants and pollinators respond differently to changes in these certain climatic cues, they may develop mismatches in the timing of their life history events. So we might have situations where there's a plant that is blooming and ready to be pollinated, but its pollinator isn't out and about and there to pollinate it yet. Or maybe the insect has emerged and is ready to be out and visiting flowers, but the flowers that it prefers haven't reached their peak bloom yet. So this graphic just shows kind of a couple of different examples of how that can happen. So we have like a generic plant here in blue and its pollinator in green, and on the left we see that the peak bloom period of this plant has shifted earlier in the season, and the peak activity window of its pollinator has shifted later in the season, so they might be responding to different cues or responding differently to the same cues. On the right, we have both the activity period of this generic plant and its pollinator shifting earlier in the season, but one is shifting much more quickly than the other. And so in both cases, that window of overlap between the flowering and the flight periods is getting smaller. We also know, of course, that some habitats like the alpine and the Arctic are particularly sensitive to climate change and can respond more strongly to these warming trends. And of course, I'm sure all of you know that historically collected specimens are very important and they can indicate long-term changes in phenology through their collection date. So here, of course, is an herbarium specimen. This is one that I collected in 2016. And there's all kinds of data on that label, including the collection date. And so assuming that we're collecting plants while they are in flower and pollinators when they are out and about and able to be pollinating, then that collection date can tell us something about the phenology for that given specimen. So the question that we wanted to ask with this research was, are there differences in the potential of alpine and lowland plants to develop plant-pollinator phenological mismatches? And we hypothesized that phenological shifts, and therefore the potential for these mismatches, would be unique in those climatically sensitive alpine communities of Northern California. So how did we go about assessing this? What kind of data were we looking at to figure out what was happening? Well, for the plants, we used collection records that spanned up to 156 years from Northern California counties. We used taxa that were well collected with a high availability of collection records and that also had relatively short flowering periods. And we sourced all of this data from CCH2. And for the pollinators, we did the exact same thing, same sort of data. We used collection records from all of those same Northern California counties, spanning up to 117 years. We used bee taxa specifically that were known to pollinate the plants that I had already selected. And all of this data came from the symbiotic collections of arthropods network, which is basically structured the exact same way as CCH2, but for insects instead of plants. All of the same types of collection data is associated and available there. So I had this big data set of all of these plants and pollinators, and we needed to constrain them by elevation to be able to draw some sort of contrast between what was happening in the alpine and lowland habitats. So the alpine records were limited to those that occurred above 2,700 to 3,200 meters, depending on latitude, because alpine occurs at different elevations depending on where in the state you are. And the lowland records were limited to those that occurred below 1,500 meters in California's foothills, valleys, and coastal regions. So then we had this big data set of plant and pollinator collection records that were filtered into alpine and lowland groups. There were, not surprisingly, considerably more records available that represented the lowlands, but we'll come back to that. For now, we'll just look at how we went about assessing these phenological shifts in those groups. So we started with some modeling. We used the specimen collection dates in the form of day of year between 1 and 365 days as a proxy for phenology. And then we modeled those dates based on the collection year to see how the collection dates changed over a really long period of time. We also threw in latitude and elevation variables into those models to control for any kind of spatial variation that was occurring. And then we used all of that to construct habitat-wide linear models that pooled the data across taxa so that we could compare what was going on with the phenology of plants and pollinators in these two different habitats, the alpine and the lowland. So finally, we were able to dig further into some of these groups, which I'll get into in a second, to construct interspecific models that would allow us to compare phenological trends over time among the taxa that were represented. And then when we constructed these models, we also included testing for independent contrast just to make sure that any similarities or differences that we were seeing were not due to relatedness among the taxa. So what kind of results were we interested in specifically? Well, we were interested in the slope values that were produced by those models. So the slope values show us how the phenology is changing over time. Here are just a couple of examples just to give you an idea. We see a couple of examples of taxa that I studied. So we've got ranunculus for plants and andrena for pollinators. On the left there is an alpine ranunculus species in red and a lowland species in blue. And then on the right are alpine and lowland andrena collections. Across the x-axis, we can see the year of collection of a given specimen between about 1920 to 2019. And on the y-axis is the collection day of year between 1 and 365 days. So what's produced is a line through each sample that indicates both the direction and the magnitude of the shifts in collection dates or phenology over time. So a negative slope means that the phenology is getting earlier. Positive slope is like later phenology over time. And what we can do is compare all of those slope values to see if the differences in those trends could be leading to phenological mismatches over time. And so this is exactly what we did for each of those 4 groups: the alpine plants, lowland plants, alpine pollinators, and lowland pollinators. So interestingly, when we looked at the alpine plants, we didn't really see any phenological change. The 95% confidence interval for those slope values, that shift, ranged from 0.1 days earlier to 0.07 days later per year. So not really too much to call home about. Similarly, the alpine pollinators didn't appear to be shifting too much, with the change in collection dates ranging from 0.03 days earlier to 0.6 days later. But when we looked at the lowland, the plants appeared to be advancing their phenology, so blooming earlier in the year, between 0.14 and 0.01 days earlier per year, which doesn't seem like a lot, but when you put it against the 156 years studied, that's as much as 21 days earlier every year and as little as 2 days. And the pollinators were shifting even more quickly, with collection dates occurring about half a day to a third of a day earlier each year, or as much as 56 or as little as 39 days over 117 years, which is a notably faster change to earlier phenology than these alpine pollinators, than their lowland counterparts. So we wanted to dig into the lowland plants and pollinators a little bit further, just to figure out what was going on with those changes. And so we constructed some models that would compare the trends among individual taxa that we studied. So this giant figure, I don't expect you to read all of those little numbers on there, but I'll give you kind of an overview. The figure shows the lowland pollinators that we studied along that left side and the lowland plants across the top, with the confidence intervals of each of those slopes or those shifts also included. And so anywhere where the rows and columns intersect and there's something in that cell means that there's a documented relationship between that plant and that pollinator. And then the upper halves, that upper triangle, indicates the phenology for the plants, and the lower half of the cell indicates the phenology for the pollinators. Where it's black, it means that the phenology is getting later. Gray, the medium gray, is no change, and the white is earlier. And so essentially what all this wraps up to is that anywhere where the shading is different between a plant and its pollinator means that the phenology is doing different things there, and there's a potential for some sort of misalignment. So one example is for bombus here, that whole row. When we look at bombus, which bumblebees are an important generalist pollinator for many plants, we see that its phenological shifts are out of sync with all but 3 of the lowland plants that we studied here. So just as an extra step to make sure that the slope or shift values that we were comparing reflected what was actually happening out in the real world, I did a little bit of field work, and I plugged my field season, which was either 2020 or 2021, into those models along with some location data for known populations. And I went out into the field on the days that my model predicted. So if you put in year, it'll give you a day. I went out on that day to see whether or not the population that I was visiting was at peak bloom. So I recorded counts of flowers that were open, senesced, or in bud, and then took an average for that population to figure out to what extent that population was at peak flower, just to see whether the model was doing what we expected it to and whether we could rely on those predictions. And what we found was that the models accurately predicted that peak flowering for 72% of all of the plant taxa that I visited. 2020 and 2021 were some messy years. There was travel restrictions associated with the university and COVID-19. We had the Dixie Fire. There was a lot going on, so there were 8 taxa that I could not visit. But I think 72% of the ones that I could is a pretty okay measure. So what does all of this mean for that original hypothesis? So to remind you, we had hypothesized that phenological shifts and therefore the potential for those phenological mismatches would be unique in the climatically sensitive alpine communities of Northern California. But we found was that contrary to that hypothesis, there was no evidence for unique Northern Californian alpine phenological changes, and that in contrast, the lowland habitats showed greater potential for those mismatches. What does that greater potential mean for lowland taxa? Well, for specialists or plant taxa that are dependent on generalist pollinators such as bombus, which we saw, or even talking about bumblebees themselves, they may be at particular risk if they are out of sync with their partners. But on the other hand, the nice thing about looking at the lowland is that there's a high level of biodiversity there. And so in areas where there is high lowland biodiversity, the effects of these mismatches might be buffered a little bit as more plant and pollinator partners are available. So this is kind of what that looks like. This is an example of the buffering phenomenon. This is a study from Bartomius in 2013, which showed the peak bloom period of an apple orchard, which is indicated by the vertical dashed lines there, centered around 0 on the x-axis. And that orchard's pollinators are distributed across the y-axis. And so what we can see is although most of the pollinators individually show some degree of asynchrony from the peak flowering period of that apple orchard, collectively the species provide pollination throughout the flowering period of that orchard. When we look at Northern California's alpine, the lack of evidence for phenological shifts is contrary to many existing studies, which document alpine plants and pollinators responding strongly, usually with shifts to earlier flowering and emergence flowering and emergence for plants and pollinators in response to climate change. So you may remember that I had mentioned that we had many fewer alpine collections to work with than the lowland, which is somewhat to be expected given that the alpine is A, a lot harder to reach, so less people are collecting up there, and B, it just covers a smaller percentage of land mass, so there's fewer records to work with. But with more documentation of alpine collections, we might be able to construct some models that are similar to that one that I showed that had all of the taxon-specific changes for the lowland. And if we can do that, we might be able to discern whether this apparent lack of phenological change is truly a habitat-wide alpine phenomenon, or if it's messier, which I expect that it probably is. And looking forward, although few studies have looked at the effects of climate change on California's alpine phenology, monitoring efforts have started in some places in the alpine to look at how phenology is changing with accordance to climate change for alpine plants of those regions, which should shed some further light on changing alpine ecology. To my knowledge, this is also the first study that's compared long-term changes in plant and pollinator phenology across habitats. And the better that we can understand how climate may affect community reorganization that's associated from phenological mismatches, the better prepared we'll be to predict the ecological consequences of that phenomenon. And with that, I just wanted to say thanks to some really kind and generous funders, to my advising committee, to some folks that helped me out with some IDing and generally being knowledgeable gems, and also to all of the friends and family members that I adopted to be my field assistants during a pandemic and said, hello, you're gonna come and count flowers on top of a mountain with me today. And with that, I will take any questions. I think we have a little bit of time. Okay. We have time for one or two.
Hi, Laura.
Hi, Hal.
Of all the places that you went to during your study, if you had a chance to go back, which one would you recommend we go to?
Dana Plateau. So beautiful. You get up there and it looks like Mars. I was stunned, and I was really thankful, especially in the midst of the pandemic, to get outside and visit some really amazing places and such a range of habitats. I was in the desert, I was on top of mountains, I went out to the coast. I got a pretty awesome tour of so many different environments in our beautiful state. So thank you for asking. One more? Maybe there's one in the back there.
Hi, great talk. I'm sorry if I just missed this, but were your plant and pollinator collections linked spatially, or were they averaged by lowland? Like, was there any link between spatial comparison in the collections?
When you say a link between...
Like, if you're comparing this set of plants to this set of pollinators, are they from collections from the same geographic location?
Oh, in the sense that they were collected from the same counties.
Okay.
But then we controlled, there was a control for latitude in there, so there's a bit of control for the spatial variation across the vastness that is Northern California.
Cool, thank you.
Yeah, okay, I think that's my time. Thank you all so much.